There you go.
That's nice.
Just started. Okay, so maybe we could go ahead and get started. There's almost 20 people already joined in, so I'm sure more people will be streaming in. Did you want to go ahead and get your screen started, and I'll introduce you? Well, welcome everybody again to the second version of the CSU webinar series in bioinformatics this fall. We are super stoked to have Dr. Renaud Berlemont from CSU Long Beach joining us today. A little brief about Dr. Berlemont. He received his bachelor's and master's and PhD from the University of Liège in Belgium. And then he continued on as a postdoc at the University of Liège, and then a postdoc at the University of California, Irvine. After which I think he commenced his position at CSU Long Beach as an assistant professor and now an associate professor of microbial genomics and bioinformatics. We're super stoked to have him here to tell us a little bit about his cool work in his lab about nature-inspired enzymes. So without much further ado, I'll let him take it away. Thank you.
Well, thank you, Aaron, for the nice word. Thank you for giving me the opportunity to present my work here. I think it's a very good opportunity to get all of us together and to learn more about what is going on in the CSU system. So hopefully you can see my screen right now. So can you give me a thumbs up or?
Yep, everybody should be able to see it. Thank you. Yep.
Perfect. Okay. So before I get started, I'd like to mention that, well, this work has been, supported by different funding mechanisms through the NSF, the DOE, and the JGI. And so that's the first thank you there. And also, I am the one talking today, but actually the work that I'm going to present has been, it's been a long process that has involved a lot of students. So all their names are here and the list goes on. I have currently 3 students or so that are working on this specific project that I'm going to talk about today. So there is Daniel, Nick, and Ben. And also this, the result that I'm going to show have been collected with collaborator here in Long Beach and collaborator in UK, Dr. Munoz and Menaz Akhtar. So thank you to all these funding and people that contributed to the story. Now, the story that I'm going to tell is actually not how it happened. I mean, I'm not going to stick to the chronology of the events, and it's just to make it, easier to follow, right? But there was a lot of stuff going on, and well, I'm going to try to make a cohesive story with all this stuff. So let's get started. So a few words about me. So as I mentioned, I'm from Belgium. You can hear I have this strong French accent, but I'm actually from Belgium, so not from France. I drink beer and I eat chocolate. No wine, no cheese. Beer and chocolate in my case. I grew up in Liège, which is a city over there, and I studied there. Pretty much my entire life as a student was in Belgium over there. And I did a master degree in microbiology, where I studied, antibiotic resistance. And then I changed gear and I moved to the field of biochemistry, and I got to study protein from extreme environments. And then it's only later on that I get interested in bioinformatics and data analytics. And basically, at the intersection of these 3 different branches of science, here you have me. And so I joined CSU Long Beach in 2014 to teach bioinformatics and genomics. And since then, I've been running the genomics and bioinformatics labs. So these are pictures of my labs. So these are essentially, well, it shows, this is a lot of computer and a lot of space to compute stuff. But we also have a small lab where we can run some wet lab experience. And I'm going to talk a little bit about that at the end of the presentation. My research focus is really about different things, right? I'm kind of a Swiss Army knife, and I have many different interests. But most of the research in my lab focuses on understanding what microbes, aka bacteria and fungi, are doing in different environments. And specifically, we are interested in understanding how microbes contribute to the carbon cycling. So how microbes can degrade carbohydrates, essentially. But we have also other projects looking at nitrogen cycling, sulfur cycling, etc. And one of the big themes of this research is really about protein evolution. And in my presentation today, I'm going to talk about these three different things, right? So how bacteria contribute to the carbon cycling and what protein they are using to do that and how these proteins evolved. Now, if we teach, if you took microbiology or genetics or biochem, most likely you've learned about these, these big pathways that explain how we get our energy, right? And how microbes get their energy. And we spend in this class, unless I'm wrong, right? We spend a lot of time talking about how the glucose, here gets processed through the glycolysis, then the TCA cycle, then the electron transport chain, etc. However, if we look at the environment, there is very little glucose that is just floating around. And even in the human body, there is not a lot of glucose that is just floating because it has to be tightly controlled. If the glucose is not controlled, then you develop diabetes. So there is no glucose. However, we spend a lot of time talking about how this glucose is going to be processed. So what happened actually is that in the environment, all this glucose and most of these other sugar are not stored in the form of monosaccharide, but instead they are stored in the form of polysaccharides. And so these polysaccharides, we can, just to name a few, we have the structural polysaccharide that we find in the plant, for example, the cellulose and the xylem. So these are very abundant source of sugar, but they are structural sugars, so they are not available for digestion, etc. So we can do nothing when we chew on wood, for example. Another very important polysaccharide across environment is the chitin, which is the second most abundant source of organic carbon on Earth after the cellulose, actually. And this chitin is very abundant in soil because it's the cell wall of fungi, but it's also very abundant in marine system where all the arthropods, essentially, they have some exoskeleton that is made of chitin, and it's just a polysaccharide. It's made of sugar. And then next to these structural polysaccharide, we have some polysaccharide that are used to store energy. And of course, the two big categories, or the two big examples, are the starch and the glycogen that are used by animals, plants, but bacteria also produce these type of polysaccharides. So in the environment, we have a lot of polysaccharides, and they need to be degraded into monosaccharides. And then eventually those monosaccharides are going to be degraded through the glycolytic pathways, etc. So if in 2024, if we just zoom in and we forget about the primitive Earth, etc., we can summarize the functioning of the Earth's ecosystem as follows, right? So we have what we call the primary producers, plant, for example, in terrestrial ecosystem. And those plants, they use the energy from the sunlight and they combine it with a pinch of water and a pinch of CO2. You shake it a little bit and then you do the photosynthesis, and the product of it is the glucose. These plants, they will use the glucose for two different purposes. First, they're going to degrade this glucose and they will get some ATP that they can use to do whatever they want to do. Some of these processes are actually using some of the glucose to create the body of the plant, because most of the plant biomass is actually made of polysaccharide, for example, the cellulose and the xylem that I mentioned before. Now, eventually, microbes evolved, right? And those microbes, they can produce some enzyme. And with these enzymes, they will attack the plant biomass in order to release the glucose. And then this glucose is going to be used through different catabolic process to generate some ATP so that bacteria and fungi can do their stuff. The focus of my research is really about these enzymes, right? The enzyme that will attack the carbohydrates, such as the cellulose or the xylem or the chitin, etc. These enzymes that we call the carbohydrate active enzyme are among the best characterized enzymes because they have many implications for biotechnology, for food processing, for health, etc. So we know a lot about these carbohydrate active enzymes. And in short, I'm going to talk about the cazymes, right? So every time I talk about cazymes, these are the carbohydrate active enzymes. If you are interested, like me, to know more about the cazymes, well, there is a database that deals with all these enzymes, and it's called the CASI database. This is a very, very important database for many scientists, all kinds of scientists, enzymologists, microbiologists, you name it. And on this database that has been cited thousands and thousands of times, you have access to all the classification of these carbohydrate active enzymes. And here, for example, you have one page. It's for the glycoside hydrolase family 5. So it's one of these family for the cazymes. And this is the kind of information that you have here. And for example, you see that the glycoside hydrolase family 5. And very often you're going to hear me talking about the GH5. GH refers to glycoside hydrolase. So these enzymes, they degrade polysaccharide. And here you have all the activities that have been associated with characterized enzyme from the GH5 family. And you have a little bit of information here and there, but actually, even if this database is central for our work and for our understanding of how carbohydrates are deconstructed across environment, there is very little information that you have here. There is no tool, there is no clear definition of what is a GH5, there is no blast, there is nothing like that. And there is not even a clear definition of what is a GH5 family. For example, we have no HMM profile, or there is no curated MSA that is directly available on the CASI database. However, remember, it's a big problem because this database it compiles all the information about these cellulases and xylanases, etc. Now, eventually, you will argue that, hey, look, here at the bottom of the page, we have a link to download all the GH5. And actually, if you download this file, this is what it looks like. So you just end up with a list of GH5, I guess. Their taxonomic origin, so these are bacteria. Then you have the name of the microbe. You have some accession number. That's good. And then you have a database. This is it. Right? This is all there is in the CASI database. There is no tool, there is nothing like that. And so this is kind of problematic because if you look at GH5, there are probably here, right? There is at least 20 different activities that have been associated with GH5. So if you identify a new GH5, how do you know what is the function of this enzyme? And so now that we understand the limitation of the CASI database, I'm going to explain you actually how it's working. So if you have a big polysaccharide, like the cellulose, it looks like this. So the cellulose is a huge polysaccharide that's actually larger than the cell itself. And this polysaccharide is made of a long chain of glucose units that are connected to each other. Eventually, the bacteria will produce enzyme, and these enzymes are going to clip in the cellulose, and the different enzyme family that are associated with this cellulase activity are from the GH family 5, 6, 7, etc. Right? So we have a large array of enzymes that can degrade these polysaccharides. And in biotech, if we just try to digest cellulose with cellulase, the end product of this reaction is going to be the cellobiose, which is a disaccharide. So it's just 2 units of glucose, and that's much easier to degrade. Now, some of these enzymes, the cellulase, right, are slightly different enzymes, right? These are actually enzymes, they look like this. So you have the catalytic domain that can clip in the cellulose, and attached to it, you have some carbohydrate binding modules, so CBM. And those CBMs, they are basically some domain that will attach the catalytic domain to its substrate. And once the enzyme is attached to its substrate, then it's going to spend a lot more time clipping in the cellulose than just diffusing around in the liquid. So the enzyme, and we have the 2 flavors, by the way. We have enzyme without the CBM and enzyme with the CBM. But in terms of biotechnology, the enzyme with the CBM have proven to be way more effective at degrading the cellulose than enzyme without the CBM. However, again, if we go back to the CASI database, there is no information for all these enzymes, right? There is no information about which contain the CBM and which do not contain any CBM. So it's kind of, again, very limited data set. Now, how do we go from a single domain enzyme, a single clipping enzyme, to an enzyme that has the clipping ability plus the binding ability? Well, most likely it's about recombination. There are other processes that have been described to explain how it goes, but most likely it's about recombination. So if you consider two genes in the bacteria, we have gene A and gene B. Those two genes, they can do their stuff and they encode each for some proteins. Now, eventually, every now and then, you're going to have some recombination. It's essentially a random process. And these recombination are going to, in some case, are going to fuse those genes together. And depending on the type of or where the recombination takes place, this eventually will lead to the production of the operon, just like the lactose operon. But in some case, the crossover or the recombination is going to take place inside the coding sequence of this gene. And it's going to produce some hybrid genes. And depending on how exactly the recombination takes place, very often you're going to have what we call a non-productive recombination. Basically, this is a recombination that fuses the two genes together, but the fusion is out of frame. So essentially, the beginning of the protein or the beginning of the gene seems to be okay. But after the recombination, the sequence is out of frame, and basically you have a non-functional protein with two most likely non-functional domains. So we are not very interested in these proteins because they are unlikely to be very good. But every now and then, you have a happy little accident, and you have a recombination that combines two domains into a single gene or into a single protein. And if you're lucky, then the blue protein is totally functional, and the red domain or the red protein is also fully functional. But they are now acting as a single gene or as a single protein. Now, because it's a random process, it means that essentially you can combine different functions. And these different functions, they need to synergize. If they don't synergize, it's most likely that the fitness of the organism where this mutation takes place is going to be reduced. And in just few generations, you're going to lose these new protein. So imagine, for example, that the first domain, the blue domain, is coding for a clipping enzyme, a cellulase, and then the red domain, it's still maybe fully functional, but it's encoding for a DNA polymerase or something like that. Then, even if the DNA polymerase and the cellulase are fully functional, it doesn't really make sense to produce this hybrid protein that is both a cellulase and a DNA polymerase. And so it's not going to be selected by natural selection. However, every now and then, it's very rare, but every now and then, we have fusion between two domains, and those two domains are involved in similar process, and it's actually beneficial for each of the two domains. And so in this case, with the cazymes, what we see is that we have some cellulase domain in blue, for example, that then fuse with some binding domain, like the little band-aid that I showed you before. The bacteria do that every now and then, and eukaryotes do that as well. So eventually, when bacteria put these domains together, this results in these very complex protein like this here. You have the blue protein that is very large here. And you see that in these protein, you have many different domains that are combined together. Now, these proteins are very large. They have many domains, but they are also very interesting because all the domains, they are working together to the same process. In this case, this is actually a structure that shows the overall structure of what we call a cellulosome. So it's a collection of proteins each with multiple domain, and the multiple domain, they are all pretty much involved in the cellulose deconstruction. And bacteria and some fungi do this kind of very large protein. And it's very interesting for the bacteria and the fungi to do these very large protein because it's putting all together different protein that will synergize and help degrade the cellulose. Now, the problem when we talk about biotech is that these protein, if we try to produce and control exactly how they work, they are getting very large protein, so they are difficult to produce. And then we have many different proteins that needs to interact together. So again, it's very difficult to control the stoichiometry of these different protein complexes. But nature has selected this type of very complex system in bacteria and in some fungi. Now, this is how we get those multi-domain proteins. If we try to understand what is the diversity of the cellulase, for example, the GH5 or 6 or 7, we can actually look at all the data that have been published. And what we did is, in my lab, is we developed some data mining or text mining algorithm that can do some text mining of PubMed. And basically, it's retrieving all the sequence or all the publication that contain information about specific families of the cazymes. And so here we looked, for example, we downloaded all the sequence for these biochemically characterized enzyme. And here, this is just the GH family 9, but remember, we have this information for all the carbohydrate active enzyme. So if we look at this information, we can do some detailed analysis of the sequence. And here you have a very, the manuscript is in preparation actually, but we have a very detailed understanding of what is the precise domain architecture of these different enzymes that have been characterized. What is very interesting is that the different GH9, eventually they show different architecture, and we can also associate different activities that are directly affected by their architecture. So having different combination of domain result in different activities. And this is a way that we can basically assign new function or specific function to specific sequence, at least in GH9. Now, if we look at the GH9, this is what it looks like. So here you have the GH9 family, and you see we retrieved almost 154 biochemically characterized sequence. And for these sequence, we were able to assign the actual function. So out of 154, 133 of these sequences are cellulases, which is a good information because it means that if you find a new GH9 in your lab, then eventually the odds are pretty good that this new GH9 is going to be a cellulase. Now, GH9 is definitely a cellulase family, but there are many other family of cazymes that are also cellulases. And so here, for example, we have the GH5 family that I mentioned before. And you see that in the GH5 family, we have way more protein, and the distribution of the different activities is different. So there is a lot of, mixed information here. And actually, there are tons of different families of GHs that are associated with the cellulase activity. So here we have all these cellulitic enzymes that are highlighted in red. If it's black, it means that no enzyme from that specific family is associated with the ability to degrade the cellulose. But you see that many different families have been identified. In GH9 and in GH5, most of the enzymes are cellulases, but some have other function. Now, in some family, for example, the GH8, the ability to degrade the cellulose is a side activity. So actually, if you isolate a GH8 and you ask me what is the function of this enzyme, I'm not going to be able to tell you what it does exactly, because it can be either a cellulase or a chitosanase or many different activity, and there is no clear consensus on what is the activity of that specific protein. So that's kind of the situation right now. Now, eventually, if we go back to the system where we have all kinds of cellulase that have been identified with or without binding domains, well, eventually this is going to release, their activity is going to release some cellobios. And one big problem that people in biotech have is that when they degrade cellulose in some application, the increase in the concentration of cellobios has major negative concentration because this cellobios can do inhibition by the product of the enzyme. And so it means that as the reaction proceeds, the reaction rate is going to decrease because you tend to accumulate the cellobios. And so that's a big problem in biotech. But if we look at how nature has actually solved this problem, it's very easy. So nature has evolved another type of enzyme that we call the beta-glucosidase. And these beta-glucosidase are going to clip the cellobios into glucose that can then be channeled through the glycolytic processes, etc. These reaction takes place inside and outside the cell, and eventually there are some specific transporters that are involved in this process as well. I'm not going to talk too much about the different types of process of transporter, but they are very important part of the story. Now, you understand, or hopefully you get a good idea of how cellulose is being deconstructed. And basically, if you understand this, you understand how the other polysaccharides are going to be degraded. So for example, here we have the xylan, and the xylan deconstruction is fairly similar to the cellulose deconstruction. The only difference is that because the xylan is a decorated polysaccharide, you need an extra type of enzyme that we call the carbohydrate esterase that is going to remove the decoration, but then you have enzymes that are going to clip in the xylan backbone, and then some xylosidase that will release the xylose. And then it's pretty much the same story for all the other polysaccharides. However, the big problem right now is that we have no tool or no available tools to look at the association between the binding domains and the other domains, the catalytic domains. So that's a major problem. And so a few years ago, it was actually before the COVID, we started working on developing a system to track the specific domain association in all these enzymes. And the system that we came up with, we name it the Gene Hunt. And Gene Hunt, first, it's a very good name, I think. But what is also cool is that GH stands for glycoside hydrolase. These are all these cellulase, etc. And so Gene Hunt was developed to identify all the cellulase, but also to identify the specific domain architectures of these proteins. And very quickly, Gene Hunt is actually based on a sequential hidden Markov model identification of the cazymes, and it works very well with different databases, with different sequenced genome and sequenced metagenomes. So here we have the detail or a more detailed view of what it does. But basically what we did is we used this Gene Hunt algorithm, which is fast and sensitive. We use it to analyze different databases. That's the first thing we did. And so, for example, we analyzed the N5NR database, which was a very large database, a non-redundant database that was used back in the day to do metagenome annotation by the NGRAST platform. And when we analyzed this massive database, we listed tons and tons of combination for the different domains. And so here in red, you have all the cazyme numbers. These are the families. And you see that we have connection, for example, between GH9 and GH8. So it means that some proteins, they have a GH9 domain and a GH8 domain. If you have a blue dot, a yellow dot, these are the CBMs, so these are the binding modules. And you see that we have a lot of different combinations. On top of that, you also have all the gray dots, and the gray dots are all kind of domains, protein domains, sometimes associated with the cazymes, but we couldn't find any reason, or, I mean, those different domains, these gray domains, they are not involved in carbohydrate deconstruction. So the existence of these associations between the cazymes and these other domains really shows that recombination is a random process, and every now and then you're going to have some random stuff that are going to pop. Whether or not these things are going to be selected by natural selection, that's a totally different question. But we created a big repertoire of all the combination of cazymes. Using the same approach, the gene hunt algorithm, we also started to look at sequence genome. And for example, here you have, this is a figure from a recent publication. And in this paper, we looked at all the completely sequenced microbial genomes from the BVBRC database. So we looked at almost 16,000 genomes, for which we have tons of metadata. And we were able to list all the cazyme and proteins. So here you have all the cazymes, and all these color-coded boxes, they represent different domains. We were also able to localize, to get the precise localization of these different proteins on the chromosomes. And we also have information about the orientation. And so we can do a lot of study about how these genes, they form clusters, or if they are evenly scattered on the chromosome, etc. And icing on the cake, because Gene Hunt was initially designed to identify the GH, but it can also identify other stuff, we were able to identify the association between the GHs here at the bottom and specific transporter that are maybe co-transcribed with the GH in order to facilitate the deconstruction of cellulose. So the idea was that if we have some gene that encodes some cellulase, maybe those genes are going to be clustered with some gene for the transport of the cellulose deconstruction product. So the expression of the cluster would support both the deconstruction of the cellulose and then the import of the cellobiose. So that was another study we did there. Eventually, we can also use the gene hunt algorithm to just, scrape data from different databases. And of course, one of the databases that we scraped was the CASI database itself. So we developed some custom code to retrieve all the sequences from the CASI, and we reanalyzed the whole CASI, and the paper is actually in preparation. With Gene Hunt, we can provide some very detailed information that can be used to link these different sequences with specific activity. And here in the table on the right side, you have, well, in the column that says CASI, right, you just have the number of sequences that we were able to identify from CASI that have been biochemically characterized. And essentially on CASI, this is the only thing you're going to be able to access, right? You're just going to have some numbers, and you know that, for example, in the case of GH1, glycoside hydrolase family 1, well, there are 340 proteins that have been characterized, and that's it. However, thanks to Gene Hunt, we can identify the different domains in these different proteins, and we know that, for example, for GH1, 300 of these proteins are just one domain. And then we can investigate what are the other domain in the multi-domain protein. And we can also investigate how the presence of additional domain affect the activity of the protein. Finally, because last week the presentation was on metagenomes, etc. I have to mention that we can use Gene Hunt on metagenomes, and actually we did. So what we did here is we downloaded this massive data set that is the Genomic Catalog for Earth Microbiome, which is a fantastic resource. And this resource was compiled by scientists at the Berkeley lab. And what they did over there, they collected tons of data sets from around the world. They assembled those metagenomes, and they did metagenome assembly. So they were able to identify almost 52,000 MAGs, right, that are directly derived from the environment. And so this is a very interesting resource. And of course, we analyzed this data set as well. What we found across environment is that essentially here on the top, we have the distribution of the different lineages across environment in green. And you see that different lineage, right? They have different distribution across environments. So you have all the different types of ecosystem here on the right-hand side. And you see that we have, for example, the gut of humans has been investigated quite extensively. And there are some lineage here that are systematically associated with the gut. We have some other lineage that are systematically associated with. For each of these mags, we were also able to identify the distribution of cellulase and xylanase and all these kind of things. And on top of that, we were able to identify the specific association between these cellulase and these accessory binding domains and other random association that are popping all over the time. So you see that here we have some, it's in the boxes, we have some lineage that are hotspot for these recombination. And those lineage, they have tons of different protein that shows different association between the catalytic domain to clip the cellulose and whatnot, and some other domains that are involved in other processes. So these are some hotspots for recombination and for finding new interesting proteins. Now, with all our work, right, we were able to compare then what is available in the biochemically characterized protein listed on the CASI database, and for example, what's available in the environment. And here we have, I mean, I know it's a lot of information on this table, but so very quickly, we have the GH1 family, and in the GH1 family, we have 339 enzymes that have been characterized. And if we look at these information, they are very essential for our understanding of what is GH1 and how it works. However, you see that of these 340 enzymes, according to the characterized enzyme, there are almost 40%, sorry, 10% of these enzymes are multi-domain enzymes. And if we look into the environment, well, we are 5% of these enzymes are supposed to be multi-domain enzymes. So from 5 to 10%, I think it's still a good match. So it suggests that what we have characterized as a society or as a scientific community is a good representation of what's out there in the environment. If it's green, right, for example, GH2, you see we have 97% of the GH2 that have been characterized are multi-domain protein, and 97% of the protein from the GH2 family that we can identify are also multi-domain. So it's a very good match there. However, all the categories that are in orange are completely off. It means that what we have characterized as a society or as a group of scientists does not represent the diversity of protein in the environment. So we have only scraped the top of the iceberg, and there is a lot more that could be investigated. Now, with Gene Hunt, we can actually identify the specific domain architecture of these different proteins, and eventually, at least for the biochemically characterized protein, we can assign specific function to specific domain architecture. Now, all things considered, and we publish a lot of these things, right? Essentially, we created a huge repertoire for all the GH and all their association with different domains, and whether it's coming from big database or directly from the environment, from biochemically characterized protein or from sequenced genome. Well, at the end of the day, we have this huge repertoire of what proteins are potentially interesting and what exists in what environment or in what genome. So this was the first, thing we did. And now I need to move on and change gear a little bit. I need to talk about those new enzymes that we identified. And these enzymes are inspired by this huge repertoire. And it turns out that in this huge repertoire of enzymes, we also identify weird proteins, very few of these proteins that are not just single domain, they have multiple domain. But they are not just any random domain. These are actually multi-activity domains. So very few of these proteins, instead of being, for example, a catalytic domain to cleave the cellulose plus a binding domain, they have multiple catalytic domains. And so very few of these proteins were identified compared to the multi-domain and the single domain enzyme. But we thought that those proteins were pretty cool. And so we made a bunch of prediction about those proteins. And for example, we identified what we call the linear pathway synergy. And in the case of the linear pathway synergy, you would have one protein with two catalytic domains, and one catalytic domain, for example, GHA here, would degrade the cellulose, and the second catalytic domain of the same protein would degrade the cellobiose and release the glucose. So essentially, this protein could be interesting for biotech application, aiming at degrading the polysaccharides and not suffering from product inhibition. So this was the first type of synergy or potential synergy that we identified. Another type of synergy that we identified is what we call the parallel pathway synergy. And these proteins with parallel pathway synergy, or PPS, these proteins, they have two catalytic domains, and one catalytic domain would target one polysaccharide, and the other catalytic domain would target another polysaccharide. However, if those two polysaccharides are physically associated with each other, essentially we could shoot two birds with one rock. And so one example of that would be, for example, a cellulase that would be associated with a xylanase, or a cellulase with an arabinase, or a xylanase with an arabinase. Because, for example, in the plant cell wall, we have tons of different polysaccharides that are all interacting with each other. So being able to degrade all these polysaccharides all at once would be very beneficial for biotech application. Finally, we have what we call the debranching synergy, or the DS type of synergy, where essentially this is the typical example here is the xylan deconstruction. So I mentioned briefly that the xylan is decorated. So basically you have decoration on the xylan. And the idea is that we would have one catalytic domain removing the decoration, right? That would be, for example, a carbohydrate esterase. This carbohydrate esterase would expose the backbone of the polysaccharide, and then the second catalytic domain would degrade the actual backbone. And so we identified some of these proteins where we have multiple catalytic domain, and we did a bunch of prediction. We were pretty excited about this. We published our prediction, and eventually we got some attention. So that was very nice. Now, the problem is that predictions are kind of cheap, right? But what we claim is that with those multi-activity protein, we could use these multi-activity protein to degrade plant cell wall to release very efficiently single sugar that we would feed to yeast and other microbes, and those microbes would then turn or produce some ethanol. And this ethanol could be used to produce biofuel that you just put in the car. So the problem then was that two birds with one rock, that's really nice. We have this prediction, but it would be nice actually to prove this prediction. Now, the problem was that we've been very good at, doing bioinformatics, etc., but we have no access to these sequence. We don't do DNA in my lab. We have no access to this DNA. But then, as I mentioned, I was trained as a biochemist. I did my PhD in biochemistry. And so I wanted to prove these or to test these predictions. And so if you have I really wanted the enzyme. And so if we have the enzyme, we're going to be rich because we can put the enzyme in different pieces of machine, and eventually we're going to get some very cool result. But to get the purified enzyme, it means that in the first place, we need to have the enzyme overproduced, which means to overproduce the enzyme, you need to have the enzyme clone. The gene needs to be cloned in some expression system. But again, we don't have the DNA. We can't even amplify the DNA, so that's a problem. And well, we don't have the sample for these DNA, so there is no easy way to get it. And I'm not planning on, resampling the whole stuff just to create or just to have access to these microbes. So it's going to be very difficult to get these enzymes. However, what we decided to do is reverse engineering. So we identified these enzymes and we decided, this work was supported by the JGI and the Department of Energy. What we decided is to actually synthesize some custom DNA that would match those different enzymes. And so we did the reverse engineering and the in silico or synthetic biology approach. And then we designed those DNA sequence. And then the people at the JGI, they were kind enough to synthesize those pieces of DNA, and they sent us those pieces of DNA in a tube. And then, well, we put those DNA sequence in expression vectors. We put those plasmid into expression system. We're using E. coli. We did the protein expression, and then we do some activity screening. Right? And we screen those different proteins. And this work was done in collaboration with people here in Long Beach, Jason Schwanz, Jocelyn Leon, and Victor Flores, and people in UK in the group of Dr. Munoz. And so we did a lot of preliminary characterization in my lab. So we identified some new protein that are very good at degrading xylem, for example, here. We also did some testing at different temperature because some of these proteins that were identified in database were actually derived, or the information was coming from, hot springs or Yellowstone National Park or deep sea vents. And so some of these proteins that we identify are thermostable. So that's also very interesting for biotech application. And then we did a lot of preliminary characterization. We tried to produce this protein, and that's a work that is ongoing right now. We also have it's a fishing expedition because we have lots of construction that we need to test, but here, this is an example of what we can get. So here we have one protein, and this is going to be very quick, where we have a GH43 domain. This is degrading the arginine in the plant cell wall that is linked with the GH10, and GH10 is the xylem. And for this, we were lucky for this protein to be able to overproduce it, but these are massive proteins, so it's very difficult to produce in E. coli. And then we did the purification of this big protein. Here you see it's pure. And because it was pure, then we were able to do tons of stuff. So one of the things we did is the crystallization of this protein, and this is, these results are still hot, right? We just got those results a few weeks ago. And then based on the crystal structure, we were able to identify the active residues. We were able to then target those active residues and inactivate some of the catalytic domain to have a better understanding of how those domains are interacting. And so here we have some very cool, very exciting results. It's more like enzymology, biochemistry kind of result. But in this protein, or at least in this protein, the two catalytic domains are used actually as binding domain, dynamic binding domain that can help each other in a very effective way. And we have some very effective enzyme that hopefully is going to be very interesting for biotech application. So in conclusion, so I talked about how we can use bioinformatics to analyze and to better understand the evolution of specific proteins, right? This is the gene hunt pipeline. And by the way, we can use the gene hunt pipeline for other types of activities, not just the cellulase, etc. We did a bunch of prediction and we published those predictions, so we were pretty excited about this. This result is also very important to make a direct connection between sequence identification and the activity. So that's going to be very interesting for metagenome analysis, for example. Some of these proteins have multi-domains, and some of these multi-domain proteins have multiple activities. And for the first time, we were able to type into this reservoir of new enzyme. And hopefully, we're going to be able to tap in this reservoir of new protein, and we will find eventually new protein for biotech application. This process of producing and characterizing all the protein is going to be a very long and slow process. We have 85 of these constructs in the freezer right now, and we are very open to collaboration, and we are open to share all these proteins. So if somebody is interested in looking at this protein, I would be happy to send it to you. And so this is going to be essentially years and years to work to come, and hopefully we're going to be able to produce more effectively biofuel. Now, as a last thing that I wanted to mention is that we use this gene hunt algorithm also to identify other function. And one of the functions that we are really interested in is the ability of some enzyme to degrade plastic. And in the coming years, we're going to start developing this exact same approach to identify enzymes that can help us that can help us identify new enzymes to degrade plastic, because we think that it's going to be another very, very important problem. Thank you for your attention.
Thank you so much. Oh, this is so cool and exciting. I'll let Scott actually lead the question answer session, because I think he's got lots of questions from his students back there.
Absolutely. Yes. Those students have some really good questions that we're going to throw your way. So Jasmine's asking, is the gene hunt algorithm concurrently checking databases, or is it searching one database at a time? And how long does it take to run a search with gene hunt?
So it's so gene hunt is going to, so the way it's designed right now, gene hunt can be implemented in other programs or other loops, right? But gene hunt is one piece of code. So eventually you can throw whatever data you want in it. So you can download some genomes, for example, from the BVBRC database, and you can decide to look at just one genome at a time. You can also embed gene hunt into a more complex program that will, for example, loop through all the genomes from the BVBRC. That's really up to you. Now, something that is very important to understand is that gene hunt, so it's a sequential screening, and it's supposed to be very fast. So let me get back to the slide. So gene hunt is fairly fast because we can, so it's actually using the PFAM database, which is a very important database containing HMM profile. And the first step is really to create a subset of HMM profile that are important for whatever function you are interested in. And so this step is very important, right? So you can specify what function you are interested in. And then the idea is that you can throw at gene hunt whatever sequence you want, right? It can be DNA or it can be protein sequence. It doesn't really matter, because if it's DNA, it's going to use, you can convert it to you can predict the protein sequence, and then it's going to use a HMM search, which is very quick, but it's not very sensitive. And the problem is that this HMM search is only going to search for the domain of interest. So eventually you're going to have some false positive. They are matching with your domain of interest, but they are only popping because you are only looking at those domain of interest. But the key is that this step is very fast, but it's not super specific, and it's only finding the domain of interest. So it means also that if you add more and more function, it's going to take longer, right? But eventually you're gonna end up with some potential positive hits. So this is what we call the PPH. And then the potential positive hits are going to be scanned then against the entire PFAM database. And this HMM scan then is going to run a very precise scan of not just the domain of interest, but all the domain. And so it's going to list and pull all the protein that are really matching with your domain of interest, but it's also going to identify all the associated domains. So the long story short, it really depends on how many domains you are interested in. And the more domains you are interested, the slower it's going to get. But overall, for example, in the paper where we analyzed, all the bacterial genomes, downloading the genome and processing the genome from the BVBRC itself and running GeneHunt, it probably take 1 or 2 minutes to run the whole process.
It sounds parallelizable too.
Oh yeah, I mean, the idea really right now is well, we are very proud of it, but this is, it's a very simple program actually, right? You have a HMM search, you narrow down the amount of sequence that you need to analyze, and then you analyze those sequence carefully with HMM scan.
Oh, that sounds really cool. Miguel is asking, can this, well, actually we had Ryan was asking, can the knowledge be used to beat bioremediation efforts? And I think you answered that question with your plastic example, which is really, really cool. So that's a great question. And Miguel asks, can this knowledge of enzymes be used to engineer easier to digest foods?
Well, so I'm really focused on, biofuel. This is one thing. But these enzymes for cellulose and xylane, etc., they of course they have a huge impact on the food industry, right? And so yes, there are tons of application for these enzymes, and this is the reason why enzymes are probably among the best characterized enzyme, right? Because whether we're talking about bread making industry, whether we are talking about pre-processing of the food for feedstocks, I mean, there are tons and tons of application for these enzymes, whether it's bioremediation and whatnot, biofuel, name it.
Really cool. Donna asks, or Dina asks, how did you find the difference between GH9, GH5, GH8? Were these based on experiments?
So this is where it's very, well, I don't see any people from the CASI team on the chat, so I have no problem talking about that. So back in the day, right, those different protein family, actually, they were identified by the CASI people, right? It's a group, it's a very famous group. They are located in France. But they identified, and they did a great job at identifying those different families. And the way they were identified or defined is essentially you identify a bunch of function, you identify the corresponding sequence, then you create some multiple sequence alignment, and you are able to identify some conserved pattern in the sequences. And then eventually you can end up defining your protein family. And this approach is very standard in the field of bioinformatics. This is how the PFAM database essentially works, right? It's based on multiple sequence alignment and the identification of conserved features in families of sequence. Now the problem is here on the, well, GH5 is an example which is very good. The GH5 family was identified a long time ago, and it is actually listed in the PFAM database. And in the PFAM database, the GH5 family is actually referred to as the cellulase quote-unquote database. And the corresponding number is PF00150, right? So if you're interested, this is what it is. Now, the problem is that here, there is no information that points to this, this GH5 family from the PFAM database. But eventually, if like me, you spend a lot of time reading and working with this, you know that. So that's only a problem if you are new to the field. Now, it's a problem, definitely, because people in the CASI database, every now and then, they come with new protein families. And for these new protein family, there is no equivalent in the PFAM database. And so when I somehow complain, right, that here there is no explicit definition of what are the different families, it's really because we don't know exactly. I mean, there is no tool, there is no HMM profile that we can download from the CASI database in order to identify these new protein family that were defined by the CASI database. So I mean, it's a very complicated, and there is a lot of things, you know, that are happening on the CASI side of life, but it's a long, I mean, it's been well
Like
described.
what you're describing in some ways is the reason why bioinformaticians are so important and how much we need people who understand biology as well as computer science. I always tell my students that none of, you know, you can do all the coding you want, but if you don't understand the bottom line of like, what are these proteins? What is a protein family? What is a domain? All this kind of stuff, you have no idea what your output means. And you can't understand what the problems with the databases are. So this is a really good example for all my students here and everyone really. We got a couple of good questions. Another question for you. How do you interpret the 3D structure information in CASI? And also specifically, what does this is more of a technical question, but what does a beta/alpha 8 barrel mean? Like, I think I understand that one, but do you know what I'm saying? It's like, yeah, okay. So those two questions.
So so the reason why, it's kind of a convoluted question, right? But the reason why we have so many different activities, for example, associated with GH5, it's a very annoying question, by the way. But one of the aspect of this is these sequences are evolving, and eventually you can have small mutations here and there that will affect the ability to bind one substrate or another substrate. Eventually, it's going to affect, you know, what the enzyme can do. And this is a diverging process, right? If you have one enzyme, it can do one thing, but eventually mutants are going to be doing different things. But overall, all the protein from this family, we can align all these proteins because they belong to the GH5 and they share the same conserved residues. The sequence of these different proteins, I mean, many of these proteins have been crystallized, actually, probably hundreds of these proteins, probably at least 100 GH5 have been crystallized. And if we look at the overall structure of the protein, all the proteins, they have this topology. This is beta alpha 8, right? So this is the overall topology of the protein in that specific family. And different protein family, they share the same overall structure, and this is what we call the clans, right? So the clan A, for example, this is the beta alpha 8 barrel structure. And in the clan A, you're going to find, for example, the GH5, but you're going to find also some, I think the GH10 are part of the clan A, and you have different overall topology. So if you want the clans that directly refer to the overall architecture or 3D structure of the protein, there are some sort of superfamily, structurally defined superfamily. But within this family, you have some variation, and then you can identify families and substrate specificity, etc.
Well, Arun has to leave right now, so we're just gonna, he has to run, so we're gonna say goodbye to Arun. Yeah. Is the beta alpha barrel is like, it's a barrel, so is it to go beta alpha, something like that, or is it?
Exact. So you have 8 repetition, and if we go to the, let me go
So
there.
it's like 8 beta alpha repeats, it sounds like. Is that what that means?
Exactly.
Okay. So that's what the 8 means. The 8 means that there's 8 of them. The beta alpha means that they're together, and the barrel means that they're shaped like a hole, like they've got a.
It's a barrel shape. So here you have the GH10. You have GH10 and it's on the side, but you see the helices and you see the beta sheets, right? And you have an alternating pattern where you have one helix, one beta sheet, one helix, one beta sheet, right?
That's cool.
It's alpha beta.
Yeah, we were just talking about that in my class, so that's really cool. I want you to know, like, me personally, I just published a couple of papers on using phylogenetic analysis to automatically take, say, PFAM families and look at the subfamilies within them that might have a functional difference. So what we can do is we can take any of those families and have a program that basically, you know, builds a phylogenetic tree and then uses phylogenetic tree plus some other tools to actually ask which of the monophyletic groups are differentiated enough that they may have a different function. So if you're interested in talking to me about that, and then what we did was we did structural analysis of the different subfamilies, looking for other things, like we did ancestral sequence reconstruction, and try to figure out if we could find new structures and everything. So anyway, it might be interesting for you to in your context.
I mean, it's very, I mean, what we did here is very similar to what you described, right? So we retrieved just the sequence, but what is nice here, I mean, first, I think that we have way less sequences than you, right? That's one thing, but the beauty of our approach is that these are only characterized proteins. So we have biochemical evidence that these different proteins have these activity. And so what we are working on right now is a system that essentially takes whatever new sequence you have and is going to do some placement on this tree. And eventually we're going to be able to assign a specific function to your new sequence.
Right. Yeah. Our idea was to do it for all protein families and then find novel ones that no one has characterized, and someone could go, figure out what they do. So
there's the, what is it called? The EFI, the Enzyme Function Initiative at the University of Chicago, if I'm correct, that is probably some very relevant or very interesting tools.
Yeah, that could be relevant. Yeah. That's cool.
Because they do clustering and subfamilies and similar kind of things.
Okay, so I don't know if you need to go. There's a couple more questions if you want to answer.
I'm here.
Okay. So what are the limitations or challenges of the known enzymes that break down plastic? Do you know about plastic degradation enzymes and what the problems are?
Well, these are just, so the two big families are what we call the PETase or the petase and the metase, right? These are different types of plastics. And so these enzymes, they are found. We have preliminary data that shows that we can identify those enzymes. These are very often some redox enzymes. So the chemistry is very different. And I'm less comfortable with redox enzyme, to be clear. But it has been done. And actually, there was a paper a few years ago in PNAS. I don't remember the author, but they created some synthetic enzyme that fuse actually one petase and one metase. And they were able to show that it's actually working. We can basically create enzyme a la carte. And of course, sometimes it's going to be a hit and sometimes it's going to be a miss. But we can take advantage of this technique where we can fuse catalytic domain in a single gene. And the beauty of it, I mean, it's obviously not perfect because we are limited in the size of the protein that we can produce. But the beauty of it is that unlike the cellulosome that is very effective for some bacteria and some fungi, we are talking about one single gene, and we know exactly what is the stoichiometry of domain 1 versus domain 2, because it's only one domain 1 for one domain 2. And eventually we can take advantage of that because it's a lot easier to engineer and to produce than multi-enzyme complexes.
And those are ones that break down plastic, you said?
Well, it was done, I mean, the one example that I have in mind is a paper that it's really about the petase and the metase, and they have some multifunctional enzyme to degrade efficiently the plastics, right?
Yeah,
different types of plastic.
Like the problem with plastics is they're like, how do you get them in solution to do the enzyme breakdown? Do you have to like chew them up first and make them?
Well, that's another very interesting question, and it's very similar to the carbohydrate because cellulose is not soluble, right? And right. So the type of enzymology that we do with those different polysaccharides is not the basic enzymology where it's not, it doesn't always follow the Michaelis-Menten model where we have, the substrate in solution and we know exactly what is the concentration. It's more complex than that. And definitely it's not my cup of tea, but I know enough to tell you that this is way more complex than just, oh, you get it in solution. Right. All these substrates are not soluble. And so we're talking about enzymology at the interface between the liquid and the solid. But it's the same for cellulose, it's the same for plastic, and it's the same for a lot of different substrates.
Yeah, I mean, I love the idea of all this remediation. I think we should just use less plastic. That's it. I mean,
Well, that's definitely the first thing, right?
yeah, I know, like
Better use it.
we got it. It's just incredible. God, awful amounts of it are washing up everywhere. It's in all our bodies. It's I actually went to a talk in Germany, and someone was studying the currents. They were taking the plastics and where they were going, and they were all going up to Greenland. So the people who live up there live in the most like pristine environment. They have no plastics. They don't have any of that stuff, and yet they are you just accumulated so much plastic there. It's wild. So one
I
more.
think it's a global problem, right?
Oh,
If you consider the giant plastic gyre, right, in the middle of the Pacific,
yeah.
it's the last time I checked that, it's the size of the state of Texas, right? And it's just
Incredible.
a huge
It's
amount of plastic.
it's just incredible. And then I remember the Philippines, they used to have as I recall, they used to have people used to carry little jars to pick up soap and other materials, and they were usable. And then they came up with these sachets, these little things like, like they would buy lots of sachets, and like a ketchup sachet like you have in McDonald's or
Yeah.
something, and they would have everything would be that, and their rivers are full of these sachets, garbage everywhere. It's so appalling to see the waste stream. Anyway, sorry, I didn't mean to go on. Let's do one more question and let you go. In terms of biofuel production, would you use these enzymes directly to degrade the cellulosic material, or did you say it would be using engineered bacteria that you would grow?
Well, it's a very interesting question, and again, the answer, it's an annoying question because the answer is very complex, right? There are many, players, there are many interacting pieces. The problem with the fermentation is that you can do it in multiple steps, or you can do it in a consolidated way, right? You can have a single organism that can be engineered so that it can degrade the carbohydrates and then do the fermentation. And there are lots of people working on developing or engineering those strains, of course. Eventually, the end goal is the production of ethanol or second generation biofuel or whatever. But these biofuel or this ethanol ultimately is also going to be toxic to the strain. And so it's really similar to the production of booze or beer or wine or whatever, right? We can get those strains to do the fermentation. Eventually, we can equip those strains with new cellulase, etc. So that's really nice. But eventually, those strains they are going to be killed by the ethanol, right? They're going to be killed by those waste products that we are looking for. And so there are also some research that is going on increasing the tolerance of these strains to higher, concentration of ethanol and whatnot. Imagine that for whatever reason, we could identify a strain or engineer a strain that would resist, instead of 15%, it would resist 18% ethanol. I mean, the whole beer and wine industry would clap, and we would
Actually,
all of us would be very happy.
Hear about the Brazilian yeast that they had, they could tolerate some extraordinary, they had evolved to extraordinary, like, I don't know, it was, I thought it was 30% ethanol, but it was some extreme amount. And at the ends of their chromosomes, they had so much of I think it was the dehydrogenase gene, like copied over and over again. It was a remarkable, I have to find that paper, but they went in and they analyzed how could something tolerate and ferment in this situation. And so anyway, it's very possible, and the yeast was doing it.
But if you put a dehydrogenase, you're going to lose your ethanol.
No, I can't remember. It wasn't dehydrogenase. It was some way they tolerated it, though. I can't remember what, but maybe I'll send you the paper if I find it again. But I was amazed.
It's a very interesting field, and I think there are lots of industry that are hoping for this kind of
absolutely. We can only drink so much, right?
Just one glass.
Yes, one glass. Exactly. Well, thank you so much. Is it Berlemont? How do you say your last name?
Berlemont.
Berlemont.
Or you can try with a French accent. Berlemont.
Berlemont? Is that close?
Good enough.
Good enough. Okay, Berlemont. I don't speak French, but And it's really nice to meet you, Ronel. And thank you so much for coming and presenting. Lots of great questions, and we learned a ton. I mean, this is, you know, slightly outside of my field, and I was like, I was amazed at what you can do with sequence data, with thinking about the problem. I'm just, you know, it's really exciting work. So thanks for presenting. We appreciate it.
Thank you.
All right. Have a good day.
Thank you. Bye-bye.
All right.